So I don't know what happened, but I'm just going to keep it pushing. Um, and so I was saying each of the macromolecules have their own bond. In nucleic acids, phosphodiester. In the carbohydrates, the glycosidic bond. And in amino acids, it is the peptide bond. So this is characteristic of uh, polypeptide or amino acid formation. And so we call the combination of these two a peptide, and then proteins are basically polypeptides or multiple peptides. Hopefully all of this is reviewed. Protein structure has four kind of stages: primary, secondary, tertiary, and quaternary. Primary is simply the sequence of amino acids, and that is enough information within that sequence in order to form a three-dimensional shape. Secondary is localized areas of structure that are formed by hydrogen bonding within the peptide backbone. The tertiary is the three-dimensional shape of the polypeptide, and then the quaternary is in the rare case where you have multiple polypeptides. So here's the primary structure, simply sequence, alanine, methionine, alanine, methionine in this case. The secondary, alpha helix or beta sheets. The tertiary is three-dimensional shape here. And then quaternary is when multiple ones get together. Tertiary is formed by interactions within the R groups of the sequence. So here, in this case, this is hydrophobic interactions. This is ionic, right? You can see that because you have positive charge here, negative charge here, and then this is hydrogen bonding. And so those help solidify the three-dimensional shape. And then these are different ways to look at the same structure. This is hemoglobin here. This is a surface model. This is a ribbon structure. And so here you can really see the hemoglobin doesn't have beta sheets, so that's a little bit weird. But you can see the different secondary structures going on, and this is called the ball and stick, which shows you the individual atoms and how they look like. I prefer personally ribbon structure because it kind of makes clear what secondary structure is. Then quaternary is when you have multiple different polypeptides that are all combining. So in this case, this is two of the same protein that comes together. So this is called a homodimer. Homo for same, dimer for two, right? Hemoglobin here is a hetero tetramer, right? Hetero, two different subunits. Tetra, like Tetris, stands for four. So it's four different units in total for it. Let me bypass this. Well, here, I'll say this. This is the sequence of amino acids here. And note that this is just amino acids. This is nucleic acid structure here. But if you look, this is insulin, and this is a sequence of insulin. And then this is beta amyloid, which is a completely different protein. And notice it has a completely different sequence of amino acids. Now, when we have mutation events that occur, right, then it's basically the same protein, but something has gone wrong in the nucleic acid structure that's caused a problem in the protein structure. So if you look at this beta amyloid, note that it is obviously completely different from insulin, but look at the mutation. Which one of these is mutated here? Um, in the beta amyloid mutation, right? This is the same, yada, yada, yada. But boom, one mutation in the beta amyloid gene, a valine to phenylalanine. Geez, what happened? Dang it. Okay, I'm not used to this. Valine to a phenylalanine, and it causes this beta amyloid is actually the protein that's responsible for Alzheimer's. So it causes a severe neurodegenerative defect here. All right, so we are focusing on the transition from RNA to protein. This is called translation. These are the players here. You obviously need the message RNA, and kind of like transcription, you have initiation, elongation, and termination that occurs, and there are different factors that are involved in there. But here, we need something that does the translation, and these are tRNAs. Well, oops, tRNAs are here. And then these are the enzymes that catalyze the tRNA production. So you have tRNA synthases, and then you have the individual tRNAs. And note with the tRNAs, they actually literally, there's this RNA that has a sequence that is complementary to DNA, and it has a region that picks up the amino acid. So this is the thing that does the translation, right? It actually looks at the genetic code of the RNA and then relates that to a particular amino acid. And they did some really cool experiments where they would change the tRNA so that it maintained its genetic code, but switched the amino acid, and they found that it would put in that different amino acid in its place. So this tRNA is the link between the language of nucleic acids and the language of amino acids. Neat stuff. And then you have the ribosomes, which is the site of protein production. All right, I don't know what's different there. So again, tRNAs actually bridge the amino acid to the nucleic acids. Here, this is a messenger RNA, and it's arranged in a triplet sequence. And so tRNAs recognize that triplet sequence, bind to it, and then add its particular amino acid to that sequence. And so if you know what this triplet sequence is, you know automatically what the amino acid is that's going to be produced. And this is a look at the tRNA. It is an RNA. It's single-stranded, but there are regions of complementariness that allow for the three-dimensional shape to form. And so here, this is the end in which the amino acid attaches to, and then in the middle here is what's called the anticodon that recognizes the RNA. So this is how it looks spread out, and this is what it actually looks like in three-dimensional space. And so again, this anticodon binds to the codon and then allows for things to start. Methionine always starts a chain, and so the first amino acid is always going to be a methionine. And so the start site here, the code that causes that binds to methionine is AUG. And so the tRNA will have something that's complementary to the AUG, UAC, right? UAC, and then it's always going to put a methionine in here. And so from this, we can get a genetic code, right? And so that we can look to see what each triplet codon actually codes for. And the genetic code is one, it's universal. So almost every organism in creation utilizes it, and they use the same language. There may be a couple of species of archaebacteria that have a different language, but for the most part, you're using the exact same language in this genetic code. The other thing is that it is redundant, such that multiple codons can code for the same amino acid. So it's redundant, it's universal, it's redundant, but it is not ambiguous. Big U, I can't spell this. Us. I don't know if that's how you spell ambiguous, but ambiguous, maybe. Which means that even though multiple codons can code for the same amino acid, there is only one codon will only code for one amino acid. And I'll show you what this means in a second. Okay, and this is the so-called Rosetta Stone, or the actual genetic code, right? And so looking at this, this is first position, second position, third position. So if we see a nucleic acid sequence, we always know exactly what the codes are going to be. So let's just say I'm just making something up on the fly here, but let's just say ACA, U, A, C, G, G, C, right? So looking at that sequence, I can always say, okay, ACA, start with A, go over to C, and then AC, and then look at A, ACA, and it's always going to be a threonine. But note, there are other ways to get to threonine. ACU, ACC, ACA, and ACG will always give the same uh, um, amino acid. UAC, start with U, then go to A, um, and then C, so UAC, it's always going to be a tyrosine. So there are multiple ways to get to tyrosine, UAU or UAC, but UAC is always tyrosine. It's never going to be anything else. And then the same for GGC. G is down here. What did it say? GG. GG here, C. So GGC over there. And this is always going to be the glycine. And so given any nucleic acid sequence, you can figure out what the um, amino acid sequence is. It's a little bit harder to figure out from an amino acid sequence what the nucleic acid sequence is because you have multiple possibilities for the same thing. All right, so that's the genetic code, and then here are the ribosomes. Ribosomes have three sites: an A site, a P site, and an E site, aminoacyl, peptidyl, and then the exit site. The A site are where things load on. The P site is where you get elongation of the chain, and the E site is the kind of allowing for the tRNAs to come off. And so this is what happens in a nutshell. In any time you have these ribosomes that are bound to the message RNA. So here's the message RNA here. The ribosomes load on at the start sequence, and then they just start going through three nucleic acids at a time and start um, processing it. And so you have here at the A site the new amino acid loading on. It actually binds, it adds to the polypeptide chain here. that's being made by reading through this information. And then in the E site, that empty tRNA kind of loads off. So tRNA loading, tRNA transferring its amino acid to the polypeptide chain, and then exit of the tRNA to allow for the next one to bind. Here it is again. Oops. So again, initiation, elongation, and termination, just like with transcription, although obviously the details are different. So for initiation, it always starts with methionine. Methionine binds to this AUG that starts the peptide chain. It actually moves over to the P site, and then in the A site, the next amino acid sequence comes. And so it reads, here is the codon sequence, GUA. And so the next thing that binds is going to be complementary to it, CUA. It brings a valine with it. So that's going to bind over here. The ribosome is going to move over, and then this methionine gets covalently attached to this valine. Now that this methionine tRNA is empty, it's going to leave off. You're going to have the chain of methionine and valine here in the P site, and then the next codon adds on. In this case, it's an arginine. And then that arginine is going to repeat that whole process. And so here the arginine gets linked onto the chain, and then you see it's been leucine threonine. And so this just goes kind of on and on until finally we hit a stop sequence. And here this is a codon that indicates stop. You have a tRNA that comes in that says stop, and that clips the polypeptide chain. The ribosome falls off, and then that mRNA is free to do it again. And so here, this is an actual electron micrograph of the mRNA. You can see it's 5' over here, 3' over here. 5' is where it starts. These are the ribosomes, and these are the proteins that are coming off of it. And you see that one message RNA can load on many ribosomes, and that you can have the kind of growing polypeptide chain as these ribosomes click through that message RNA. And then once it gets made, then it gets processed. It can go to the rough-ER if it's something like a membrane protein, in the Golgi to go outside of the cell. It can be made in the cytosol directly, or it can be kind of pushed into other directions. And, oops, oh shoot. Oh shoot. And so here, this kind of tells you all the possibilities. If you have a protein with 62 amino acids, and there are 20 different amino acids, then you have 20 to the 62 possible sequences here. This is equal to 10 to the 80, which is the total number of electrons, protons, and neutrons in the entire universe right now, right? So lots of possibilities that occur from here. And then, since this is neat, this is how we get into how we change and how things are different. We'll talk about genetics next semester. But here, if you have the protein sequence GONE, right, you can look and see, well, any one of these can be shifted, right? GNNG, GNGJ here, E to J. So most of these are going to be deleterious, or they're not going to form anything useful. But every now and then, you get something that forms a word that makes sense. So if you have GONE, and then this G gets transitioned to a D, then you have done. Now that word means something, and it has a different meaning than the original word, gone, in this sense. And it's the same thing in protein structure. Most of the times mutations are going to be deleterious, but sometimes you'll do something that has meaning, and that meaning actually increases the adapt, um, um, adaptation of the um, species. Whew. All right, so here we are. This is what we've done. We've started from DNA. We've transcribed that DNA into RNA via the process of transcription. That RNA is processed and then goes out to the cytoplasm where the mature messenger RNA links up with ribosomes to form polypeptides, and these proteins go out and do what it do. So that is the central dogma. DNA transcribed into RNA, RNA translated into proteins. Proteins are the things that actually do the action. Thank you for your attention. That